Okay, so today we're going to really just kind of summarize in a larger context everything that we've been discussing throughout the entire course. And I feel like some of these topics can be integrated throughout the lectures for each level of gene regulation that we discuss. So it's one of the things that we have thought about for a long time to incorporate throughout the lectures. That way it's not like you're going through all the material and then towards the end, you have all these amazing tools that you can use, and rather integrate it earlier in the lecture. So you guys can let me know what you think as well about this after. But I think it's a really exciting topic because it's really relevant to the research that we do now, or in molecular biology or cellular, cell biology, or any other type of life sciences. It's really relevant because those are the approaches that we're using a lot of. So we discussed earlier, looking at the cell as a whole, right, and the different levels of gene regulations. We talked a lot about DNA, DNA replication, how you can clone pieces of DNA, how you can identify certain features in DNA. Then the next level of gene regulation, which we talked a lot about, was also transcription, right? And not just about making the transcript, how, at least in eukaryotes, you need to be able to even access the DNA to even transcribe the region that you need to transcribe, right? And that's when we looked a lot on chromatin level regulation, right? As you can see here, right? So you need to be able to even access the DNA to be able to perform transcription. Once you have your transcript, right, you need to have many different steps that needs to be processed in that transcript to make sure that you're going to make a protein that represents whatever is coding in this DNA, and a functional protein as well. And we went through, splicing, capping, polyadenylation. We talked last week about some of the steps that goes beyond that, right? How do you get, what happens as the mRNA is going out into the cytoplasm to be translated, right? And that there is some regulation that's happening there as well. So mRNAs that are not perfect will get degraded by this nonsense mediated decay pathway that we talked about. And these mRNAs are checked during that pioneer round of translation that we discussed, okay? Once that mRNA looks good and it can be translated, then you have translation. We discussed the different steps in translation: initiation, elongation, and termination. And then even after you make this protein, you will need to be able to modify that protein, right? So have it folded properly. And in addition to folding, you can have other types of modification that's important for the proteins, such as phosphorylation. You can have modifications, methylation, acetylation, and that's where you have those proteins that, particularly these histone proteins, that are important for regulating the chromatin structure, right? So these type of modifications and proteins needs to get back into the nucleus to play a role in chromatin compaction, right? And then we also mentioned, which is not highlighted here, that you have features within the protein as well that tells the protein where to go, right? Where am I going to go? And for each of these steps of regulation, we are able to study it because we know what the structure of a gene looks like, right? We talked about this also very briefly at the beginning of the course, where we looked at this typical gene structure, but this is more on a global scale, where you can see that you have your gene structure. This is all, again, all DNA, everything every protein, the final protein product has been encoded from what's written in the DNA, okay? And within that DNA, you have many regions that are important for producing the information that's needed to be transferred to a protein product. We need, you guys recall from the level of even transcription, right? You need to have features within the DNA that helps the transcription machinery to recognize where you need to start transcription. And these are elements within the promoter where RNA polymerase, with the help of all these other general transcription factor, will recognize and begin transcription. And then you have other elements in the promoter. Again, this is all part of the DNA. It's already there when you look at this DNA sequence. Regions that are important for translation. So in eukaryotes, that Kozak sequence that you see that's present in the mRNA, that's also already embedded in the DNA, right? So when it gets transcribed, those sequences are recognized by the ribosome as it's scanning the RNA. You have also the codons that the tRNA will recognize, right, within the DNA, again, encoded in the DNA, transcribed and recognized by that machinery. Regions for splicing, and also regions downstream of the translation termination site. We didn't talk a whole lot about the 3' UTR region. It is important for regulating gene expression, but we didn't talk a whole lot about it, and it's not an area that's really well studied. We know to some extent different lengths of 3' UTR can be important for regulating gene expression. We also know that different elements reside within this 3' UTR that helps to regulate that transcript stability, for example, and also translation. And then we talked about the polyadenylation site, which gets added at the end of the mRNA after it's cleaved. So all of these features, besides the polyadenylation, which actually gets added at the end after the transcript is made, all of these other features are really embedded within the DNA. But there's also other sites that are revealed once the DNA has been transcribed that are recognized as important sites for translation and protein function later on. Okay, that's not clear when you look at the DNA that they're there, but once the transcript is made and you look at the transcript, they can be recognized by certain types of proteins. And then also within this structure, what's important is that you have these exons, right? I mean, most eukaryotic genes have exons and introns. Some genes don't. Some genes are just all exons, right? So that information is not there. And it just depends on the organism. It depends on the gene. The size of these introns and exons will vary across organisms, across genes, across gene families. So there's really no set of rules as to how long introns should be, or how long an exon should be, or how many introns should be within a gene, right? These are all information that's really just embedded within the DNA that that's part of this coding region, or this coding information for proteins later on. And so when we study the entire genome as a whole, we refer to this as a genome or genomics, right? So we're studying the complete set of genes in an organism, right? So all the genes that are there, all the pieces of DNA that's there, right? We're looking at within the genome, all of the proteins that are there. We're looking at everything within that genome. Proteins, RNA, DNA, everything. That's the whole genome. We can dissect that and say we're looking at only the transcriptome, right? So those are only RNAs, okay? Those are only genes that have been transcribed. So we're looking at all of the transcribed region within the genome, all of the mRNAs. Most of this is messenger RNA that we're looking at. And this has changed a lot. So you can also do a transcriptome. You can also look at a transcriptome to look at other types of RNA, like ribosomal RNA or tRNAs or small RNAs. Okay, you can also do that. But most of what we study when we say we're looking at a transcriptome is really messenger RNAs, which are the products for that codes for the information that you need for a protein. Okay. And we can also look at the proteome, right? All of the proteins that have been produced within that organism, and you can isolate only the proteins, okay? You know that you can have variations between the transcriptome and the proteome depending on alternative splicing, right? So if someone might be interested in seeing what are the final protein products that are functional within an organism, then you would look directly at the proteome. Looking at the transcriptome is not going to give you that information. It's going to give you an idea of, what are the genes that are being transcribed, but how many of those transcript actually make it all the way to a functional protein, you can't really tell that by just looking at the transcriptome. So people will look at the proteome to see what are the final abundance of those proteins in the genome. So the first genome that was sequenced was a phage, and really bacteriophage. It really was relatively small. It was sequenced when Frederick Sanger developed this chain termination method of DNA sequencing. It's 5,000 base pairs, which is relatively small. You can do that. You can sequence that within 5 minutes these days, right? So, but at the time in the 70s, it was a huge undertaking to sequence this 5,000 base pair genome of this bacteriophage. And he used the method that we've discussed early on that you guys are going to remember, because you're going to learn how to read a sequencing gel. We'll also need to know what the components of a sequencing reaction is going to be, right? And that's what you can see here, right? You have your DNA, you have your primers that you need that's labeled, polymerase, other nucleotides. What's critical for a sequencing reaction is to have this dideoxynucleotide present, right? Because you need to terminate that chain. There's no OH, so you terminate that chain when you need to have it there, okay? So this is how it was sequenced using this very similar method that we discussed to identify what's in the genome. Of course, later on, and I'm reminding you guys about this because it's something that you obviously will see on the final exam again, but later on now today, we're using a more automated approach where we're using fluorescent labels rather than radioisotope labeling. So it makes it much faster. You can sequence much more. You can also do this, pool all of your DNA or whatever you're sequencing into a capillary that gets read very quickly by scanning. So this is kind of the method that we use today, but it's the same information that you would get from looking at a gel. And the information that you get from doing these whole genome sequencing approaches is, I mentioned, you get the coding regions, you get the introns, you get the promoter regions, you get the end of that coding sequence, right? These are all information that you get. You get also whether or not there is mutations within the DNA, right? But in order to do that, of course, you need to compare it to something else that you know, right? But you can get single nucleotide changes in the genome by sequencing the whole genome, because you're sequencing every single nucleotide that's present within that genome. You can also get, if you got information about the relationship among genes, how compact that genome is in terms of how many genes are present in the genome, how close to each other they are, what the length of the genes are, how many introns they are, how long the introns are. So you can really get a good sense of everything that's within the genome of an organism, and if you're thinking about looking at the genome is going to be the same in every cell, right? So your genome in your liver cell is going to be, or your DNA sequence in your liver cell is going to be the same DNA sequence in your brain cell, right? It's just one genome and one set of DNA, okay? What's going to vary is expression of genes that are in those particular cell types. The proteins that are produced in those particular cell types, those will vary, but your DNA content is going to be the same. So how do you recognize a coding region? So you say you go outside and you see this unusual organism and you decide, okay, I'm going to sequence it, right? I mean, it's like $1,000 to sequence the genome. You might have some money lying around. You want to know what it is. So you can either order a sequencing kit or you can send it to a company and they will sequence it for you. And now you want to see what's in that genome. So you need to recognize what is coding, meaning what codes for genes, and what is non-coding, right? Everything else that's in the genome that's present. And the way that we recognize usually what's in a genome is by looking at particular regions or sequences in the genome. The most common way to identify what is coding in a genome is by identifying ORFs, or open reading frames. And these open reading frame is just called open reading frame because it's a region where there's no interruption in between, okay? So these open reading frames are very unique in the beginning and the end of that region. So ORFs often start with ATG, right? And you guys know ATG because it's the start methionine in most cases. And occasionally you can have GTG present within the genome, or in bacteria, you can have the two other types of start codons that we discuss, okay? But usually it starts with ATG. And this ATG is what corresponds to the initiation codon. And then you have the end of that ORF, which is recognized by the stop codons, okay? So you're starting with ATG and you're ending with one of these particular stop codons. And this is really what you're doing. You're going through that genome, trying to find these regions where you have a start and you have a stop within a certain frame. Okay, so it has to be within the same frame. We talked about the 3 different reading frames. So when you get the genome sequence, you're doing 3 base pairs at a time, reading across those sequences, looking for a start and a stop. Okay. If you, for some reason, shift frames by shifting by 1 or 2 bases where you're starting, you're going to be in a different frame. So you're going to encounter a different codon, for example. Okay. And this is exactly what you can do if you look at all the different genomes when that's being sequenced. So, and this is something that's actually Sanger discovered when he was looking at this bacteriophage, is that you're expecting all of the genes are supposed to be present and functional in the same reading frame. But what he also found was that you had these genes that are overlapping somehow, okay? And they're present, and you can identify, if you look in a different frame, do you encode a different gene, okay? And that's exactly what this sequencing the whole genome enables you to do, right? So in this case, you have this gene, you have your start, you have your stop. Embedded within here, right, is this ATG. But if you notice, this ATG is not within this 3 codon, right? It's a different one. So it's like you're shifting it over by 1 base to then be able to read this frame. So then you can start and then you will stop, okay? And it's the same here, right? It's not within this normal frames, it's in a different frame. So you can do that into the 3 frames and see whether or not you have an ORF, because if there's a different frame that's present, then it's quite likely that some of these proteins, the mRNAs from these different frames are the ones that get transcribed, you'll produce a different protein, and that protein might be important, right? It really just depends on the organism. Okay. So you can come up with these what we call genetic maps, right? The identity of the ORFs, which encodes the genes, so you know where the genes are, how many genes are there, other genes that you can identify from looking at different frames. You can identify introns, you identify the promoter regions, the elements within the promoters. All of this information you get from looking at the genome sequence. And when we initially started genome sequencing, right? Imagine you're starting from, we have PCR, right? You're starting from this little piece of DNA that you're doing a PCR reaction for, and then you will sequence it. With whole genome sequences, you can imagine if you have a very large genome, or even for that bacteriophage, it's 5,000 base pairs. What you need to do for these large pieces of DNA that needs to be sequenced is that you need to fragment them. The genome sequencing technology allows you to sequence with some confidence, or the traditional genome sequencing technology allows you to sequence with some confidence 800 to 1,000 base pairs. You can get really good quality sequences for. Now there's newer methods which can do much longer reads, okay? But traditionally it's 600 to 800 base pairs of really good sequence. So if you have the human genome, which is 3,000 million base pairs, you can sequence all of that in one run, right? So you need to chop it up into pieces. So you need to fragment the genome. And you fragment the genome by using restriction endonucleases because they will cut randomly in the genome, right? So you would do these, digest the genome, maybe with a 6-base cutter, and sequence those pieces. Before you sequence those pieces, you need to make them into clones. So you need to put them into a vector, right? You can't just randomly chop them up because the sequencing machinery need to know where to start. So you put them in a vector, and that way you can know where in the vector you can actually start sequencing, because you can design a primers to the vector. But if you randomly digest the genome, you don't know where to design a primer for because you don't know what the pieces are, right? With all of those millions of pieces that you have. So you need to take these pieces and make them into a random clone. You put them into a cloning vector. And then you sequence those cloning vectors with those little pieces. So you have all these little pieces now. What do you do with these little pieces? You need to now assemble them back into a full genome sequence, right? You need to assemble them back into the chromosomes, right? You need to put them where they belong in the genome. And to do that, one of the first things that you will do is create what we refer to as contigs, right? And contigs is just a set of clones that are contiguous, right? So they're not interrupted. So you create these contigs, right, for different pieces. And once you have these contigs, then you will look at the contigs and try to align them. The end of these contigs will have sequences, because again, it's a random process, right? So at the end of these contigs will have sequences that are similar between contigs, right? So you can align them based on the overlapping contigs. So in this case, the end of this red contig has some similarity with this purple contig. So you know, because they're overlapping, it's quite possible they belong next to each other, right? So that's kind of how really you go about aligning these genomes. This is a big problem for genomes that have lots of transposons, because remember, transposons are repetitive DNA, right? So it can really make you assemble the genome incorrectly because you have so much repetitive DNA, and you might think, oh, this is overlapping with this region, so it must belong there. But it could also be overlapping with many other regions in the genome because the transposon is going to be in other regions in the genome, right? So that was a challenge with genome sequencing to begin with. So it took us a long time to be able to figure out how to appropriately assemble genomes. Now, what we do is we get rid of all of the transposon sequences because we know what the sequences of transposons look like. So if you were to sequence this genome, you took your random clones, you would run this DNA through a program. The program will identify sequences that are transposon sequences and get rid of it. So then when you're doing your assembly process, you can be somewhat confident that you don't really have a whole lot of transposons in there, okay? So there's two approaches that we use that's kind of built on what I just discussed. And that approach is mapping, then sequencing. right? So that's clone by clone sequencing. So this approach is was kind of one of the desirable method because it also makes the assembly easier, okay? So, but in order to do this, you need to have some information about the genome beforehand. So you need to have some kind of a map, right? So this map, which is called a physical map, gets generated because you knew beforehand certain regions within this DNA, where they were located on the DNA, so you can put them together. So here you're taking these large clones that you just chopped up and put into the, that you digested the genome, you're making your clones, you put them on this physical map so you know where they belong already, yeah? And these little dashed lines that you're seeing, which are molecular markers, could be anything. It could be genes that you single-handedly discovered from characterizing a particular gene. So you know that this gene is located at this position on this piece of DNA. Quite often, these markers can also be restriction enzyme sites, right? So these can be, you know that a restriction enzyme is located on this region at that location. They can also be what we call SNPs, or single nucleotide polymorphism. So a region where you know that there's a base pair change in this particular region, right? So it's just something that you know. So we call these molecular markers because they're part of the DNA, right? So you know that they're there and you know where they're located. So you take these clones, you assemble this full physical map, so you place them already on this physical map. So now when you decide to use these regions, so you align them to the map, you take these selected clones that you just aligned to the map, divide them further into subclones, so you digest them even further, you sequence the subclone because they need to be small, and then it's much easier for you to assemble them because you knew where these came from because you already had this physical map, yeah? So you're mapping the clones to a physical map first, and then you're sequencing. You're sequencing each clone, you know that this set of clones belong here because you already put it there because you had all these markers, okay? So this is kind of one of the ways that we can really sequence a genome with an easier way to assemble it, okay? So, sorry, so yes, yes, Sheila.
I'm not quite understanding what step 2 is, like select a clone of minimal something.
So the minimal tiling path is really just the minimum region that you need in order, that represents this physical map. And because when you assemble this, it's like, imagine you have a whole chromosome. So this could be a whole chromosome that you have assembled based on the markers you know. So let's say this is chromosome 1, okay? So then in chromosome 1, now you're looking for regions. You're going to select clones to sequence on this region 1 that represents a good amount of sequences that you can be sure that if you digest these sequences, you can align them back, like here, okay? Because you will have a certain number of markers. So this is the physical map. If you take the sequence, let's say you have 7 molecular markers here, right? You digest this clone, you know, okay, with these amount of pieces, with these 7 molecular markers, you know where this came from. You should be able to put it back once you sequence it together. So this step is really to determine.
So it's just like selecting a length.
You're selecting a region, right, which is rich enough in markers so that when you're ready to assemble them back, you know that it's going in the right place.
So if we have I'm sorry, I'm just kind of confused then. If we have the clones, wouldn't the physical map already be the sequence, or?
So if you have these clones, they're not going to be the sequence because you're going to have a lot of gaps in there, right? And these physical maps are usually pieces of DNA, as mentioned, they can be genes, right? So you can imagine that you individually decide that you want to study a particular gene and you've sequenced that particular gene, okay? So these little clones here doesn't represent all of the DNA that makes up this physical map. Even though it looks like it's tiling, there's a lot of unknown that you don't know here, right? What you know is regions where these markers are. So there's a lot of gaps that we don't actually know what they are, right? But we know that, okay, we have these sites which are represented here.
Okay. That makes sense. Thank you.
Yeah. So this is really, you're trying to sequence the whole genome and see what all of the sequence is. But we already had beforehand some of these molecular markers that we've identified from using different ways or different sources. So you can kind of create this kind of physical map to place them where they're present, okay? So this is just placing them. This makes it seem like these are directly overlapping, but you also see that you have some of these gray marks which are not represented here, right? So it's really just placing the information we know, creating a physical map, selecting a region on that physical map which covers a certain amount of DNA region, and then sequencing that with the idea that once you get these little clones that sequence your 600 to 800 base pairs, you're going to be able with some confidence to put them back together because you know a lot of information already about this region. Does that make sense?
Yeah, it does. Thank you.
And this is in contrast to, for example, so this is really just saying what I pretty much said, right? So physical map. And this is really in contrast to what I told you previously, right? So you need physical maps or maps to order, overlap, orient the little pieces that you have in the genome. This is in contrast to really the other approach, which is you sequence first and then you try to put everything back together. So with the previous approach, you're putting it together beforehand based on information that you know, right? And that can be, again, restriction enzyme site, it can be genes, they can be other types of molecular markers. So you're kind of assembling like a scaffold, yeah, some parts of this chromosome that you need to sequence, but there's a lot that's still missing, yeah? So once you do that, then you can digest that or cut up that pieces, sequence it and put them back together. And it will be easier for you to put them back together because you can have regions where there's some overlap. So you know, okay, these two might belong together. And you can just assemble it that way. Yeah. So it really makes it much easier to sequence whole genomes that way. The other way, shotgun sequencing, you start from here. So you just digest all of the genome, right, into all of these little pieces, and then you try to put it back together. That's way more tedious to do, and it takes much longer to create those type of genome assemblies. So in this case, you have your contigs, which are they're small. You first need to go through this first step of aligning these little pieces together, making a contig, and then trying to hope that you have regions that are overlapping between the contigs, right? And that's the idea, is that you have these regions that are overlapping. But a lot of times you'll have gaps that's present, right? And then you need to figure out how to fill in these gaps. So that takes another level of either sequencing or analysis to fill in the regions where there's gaps, because you might not have the end of this blue or this green overlapping with this end of this purple, so you can really say that they belong next to each other or side by side, right? But this helps if you're doing it this way, because you already know, based on this physical map, where these pieces belong, right? So that gives you an advantage. It's very easy to fill this in very quickly and say, okay, all of these clones belong here. And then you just need to focus on now what's missing and then fill in the blank windows, which is much easier to do. So those are two approaches, mapping then sequencing or shotgun sequencing. So you sequence first and then you map later. Okay.
So are the two approaches complementary to each other, or like they're separate methods?
They're separate methods, but they can be combined, right? So depending on the genome, they can be combined. Depending on the genome you're sequencing, I would say almost most eukaryotic genomes, we have information about molecular markers. We have some sort of a physical map already existing. Very few genomes we don't have, yeah. So we have some idea already, and that makes it easier to then just, if we know we have a physical map already, then we don't need to go through and do this particular step. We will just go to shotgun sequencing, because then you can just align it to that physical map that's already existing. So it depends on the organism, it depends on the genome that you're looking at. This is easier for genomes where that's duplicated. We didn't talk about genome duplication. I don't know if you guys spoke about this in genetics. So you can have multiple genomes, right? Your entire genome can duplicate. So all 46 of your chromosomes can completely duplicate, right? In the genome. This can happen, and this happened in many organisms. This happened where they had a whole genome duplication. In that case, having this physical map is definitely much easier and preferable than digesting two genomes and then trying to put everything back together, right? So there's definitely, it really depends on the genome that you're looking at, and what you know beforehand about that genome will determine, which of these methods that you use. If you were to sequence a human genome, like if you want to sequence your genome now, you would just do this shotgun sequencing, because we have so many fully sequenced human genomes, and those fully sequenced human genomes are the ones you will use as your physical map, as your reference to assemble things, because you already have a lot of information. A lot of genes are shared between organisms, right? We will have genes that are similar between the two of us if we sequence our genome. We know where they're located in that region, right? So you can use previously sequenced genome as your reference or your scaffold for sequencing. So this is the method that you would use for now if you're going to sequence the human genome for anyone, because you already have so much information about certain features within that genome and certain locations and markers that you should be looking for. But if you were to, go to, I don't know, Mars and discover a completely new species or a completely new organism, then you might not have any information beforehand for that. So you might want to start trying to see, can you find certain features that you can look for? Yeah. So this is kind of the method to create those little the clones that you need to sequence. As I mentioned, you can't just digest genomic DNA and send it to sequence, right? Because you need primers, right? The sequencing technology is built on primers, so you need to know where to start, where the primer should anneal to. So you can use, and you want to clone all of those pieces, right? So if you have a large genome and you have all of these multiple, hundreds of thousands of pieces of DNA that you need to sequence, you need to put it somewhere, okay? And usually there's vectors that you can use to put your pieces of DNA. These vectors, the most common one that's used was isolated from bacteria. So this is bacteria artificial chromosomes, or BACs, right? You can insert large chunks of DNA into these BACs or vectors. Again, they also have restriction sites. They have an origin of replication. Very similar to the cloning vector that we talked about. It's just that they can really take up large pieces of DNA. So they can take up, about 150 base pairs. So you can use these vectors for cloning. So create your clones and then sequence, because now you know, okay, if it's located in this region, you just design a primer somewhere here, and then you can sequence across that plasmid. Yeah, so it's much easier for you to sequence your clones that way. And then you can also use yeast vectors, which are YACs, or yeast artificial chromosomes. These are much more difficult to use. They're less efficient, they're unstable, and then they're also difficult to isolate them from yeast. So the process or the protocol to isolate plasmids from bacteria and yeast is a little bit different, because you know, the cell membrane and everything for yeast is a little bit more sturdier than bacteria. So it really takes a little more, it's a more intensive process to isolate a plasmid out of the yeast cell once you're ready for it. So the most, I mean, it's in the early days when we were doing genome sequencing, many people were using YACs, but BACs is the preferred choice for sequencing that we can use now. Since we started from the first phage, right, this bacteria phage that was first sequenced, which I told you was like about 5,000 base pairs in 1977. A lot of genomes have been sequenced. This is actually so old. I need to update this because I think we have genome sequences for way more than this. I think we're probably in the thousands, how many genomes we have sequenced. But this just shows you the size of the genomes that we were able to sequence. They're much larger, right? The human genome is 3,000 million base pairs. And the year that we were actually able to sequence it. So all these years, you're building upon the methodology and also you know, your mistakes along the way to be able to fully sequence this human genome that was sequenced in 2010. So all of these serve now as a scaffold for any other genome within that particular species that you want to study, right? So if you want to study different mouse varieties, you already have the house mouse that has been sequenced. So you use that as your template or as your physical map or stuff like that. So you can always use that information, okay? And this is just looking at genome sequence. You do this, you know, it was, again, incredibly expensive back then. Now it's pretty cheap. It's $1,000 or probably less now. I haven't checked recently, but you can just sequence your genome and get the information that you need from your genome. So when you want to look at the functional regions within the genome, so as I mentioned, you're going to sequence your genome, you're going to have all of your DNA. It's going to tell you where your genes are, where your promoter regions are, give or take, where the introns are, how similar two genomes are to each other. Within a genome, what is the gene content and how compact that genome is, right? What it doesn't tell you is which genes are expressed. You know that there's genes, right? You know there are genes because you can see it. You can see your ORFs, right? Your ATG and your stop codon. So you know that these genes exist, but it doesn't tell you which genes are expressed. So in order to do that, you need to do a different type of sequencing. So the genome sequencing, you're isolating DNA and you're sequencing that DNA. If you want to see what's expressed, right, what do you have to use instead? What would you use if you want to see everything that's expressed in the genome? What would you use? Like I told you, okay, here's this plant. I want you to tell me all of the genes that are expressed in this plant, and I just give you the plant. What would you isolate from that plant to be able to tell me that?
It's RNA?
Right, exactly, right. So you need to get all of the RNA from that organism to figure out what genes are being expressed, okay? And the way we go about doing this, isolating all of the transcripts of that organism. So all of the DNA that has been transcribed and processed, meaning it's went through already the splicing and capping and whatever. So all you have is the stable messenger RNA that's left. You need to isolate that, okay? And that tells you that these are the mRNAs that represent that particular gene, and assume that they're going to be functional, okay? So that is referred to as a transcriptome, right? So it's all of the transcripts in the genome. So you combine those two words, right? And the way we refer to the study of the transcriptome is transcriptomics, right? So you might hear this word being used quite loosely depending on where which lab you're in, or certain material you might be reading. But it's really the way for us to study all of the expressed genes within the genome, and to also measure how much in the entire genome of that messenger RNA is present. In our previous discussions, right, and when we were going over transcription, we talked about methods that we can use to just look at a particular gene, right? When we try to be studying gene expression, just looking at a particular gene, how is that particular gene expressed, and where it's expressed, what time in development it's expressed, right? And we talked about the being able to do that in the previous lectures. So those methods where you're looking at just a single gene is what we refer to as a small scale method, because it's really small scale. You're looking at one gene or two genes, or maybe even three genes, right? But it's a small scale that you're looking at. And some of the methods that we discussed for looking at monitoring gene expression on a smaller scale, we discussed 3 of them, not a whole lot extensively, but so maybe 2 more so than 3. Do you guys remember what those are? Northern blot, yeah, and qPCR, right? So those are 2 small scale methods that we can use to look at individual genes if we're interested in studying those. And I have written here also RT-PCR and qRT-PCR. Anyone know the difference between these 2? Right, so the qPCR is quantitative, right? So you use the qRT-PCR because you can quantify how much of that gene is expressed relative to something else. RT-PCR is the same idea, except it's more of a qualitative assay, right? So you run a gel, it's present or it's not present, or it looks like it's present more than something else, but it's really a visual type of assay. You can quantify the gel after you ran it for RT-PCR. It just requires using certain technologies, and it's usually not super reliable. So people prefer to see a method that's more quantitative because you're comparing this to some other gene in the genome that should be expressed at the same level, right? So some kind of housekeeping gene or something you're measuring, comparing that to. So 2 of the larger scale methods that we use for monitoring gene expression are microarray and RNA sequencing. I like RNA sequencing as a method of choice versus microarray, and I'll tell you why that is in a minute. But these are the 2 methods that were developed to look at the transcriptome for any given organism. So with the microarray approach, the way that this works is that, and it's referred to as a microarray or a microchip, it's really a chip, like a little device. You can see it's 3 by 1, right? It's really a really small piece of device, and on that device are DNA that has been printed onto that chip. Okay, so all of these little, which you can't really see it very well, but little dots represent spots of DNA that has been printed on to this chip. And this DNA that's on there represents all the DNA in the genome, okay? So I mentioned DNA here, but obviously because we're looking at expression, this can definitely be cDNA as well, right? So you can have DNA chips or you can have cDNA chips, and you put all, again, spotting all the pieces of DNA that's present within that particular genome on these chips. This is done by using a printing technology, which actually you can, spot them a certain distance on these really small chips. And after you spot them in a solution onto the disk, they're air dried. They're covalently attached by UV radiation. So you put them in a microwave, a special type of microwave, expose it to UV for a little bit, and it will bind, cross-link that DNA to that chip, right? So bind it to that chip. And that's what you have. So that's a microarray. It's a little device. You can purchase it from a company that represents whatever genome you're interested in. And you can then use it to look at whether or not a gene you're interested in is expressed or not. And
So
the way that you
Professor,
do that, yeah.
Can you go back to the previous one? So each spot on this chip has a specific gene segment. Not all DNA,
a
right?
specific piece of a gene, yeah.
Oh, gotcha.
Yeah. So the way you use those chips is that if you now are interested in whether your gene is expressed or not, you isolate RNA, right, from the genome. You quite often, these microarray are used because you're comparing two things. So you want to see, is your gene expressed in response to a certain condition, in response to a certain chemical, in a line where there's a mutation versus not a mutation. So you're kind of comparing two things together quite often when you're using these microarrays instead of just one sample, right? You can just use one sample, so you can just isolate all of your RNA from one genome and label it with the two different labels to see whether or not it's expressed or not. But quite often you're comparing two things together for these microarrays. So in one sample, you would collect from a normal person, right? You convert the messenger RNA, because again, it's all expressed, to cDNA, and then you add a label at the same time that you're doing this reverse transcriptase experiment. You're adding a label or a fluorochrome, which is a fluorescent label that will fluoresce red when you run it through a machine, yeah? You collect the sample from maybe a mutant or somebody, some other person that you've exposed to some kind of treatment or chemical. You isolate that same mRNA, and you do the conversion to cDNA. You add a different label, right, to be able to distinguish between the two, okay? And then you will hybridize that to this chip, because on this chip, you have regions of that DNA for all of that genome, okay? So if they're expressed, then the cDNA, what's missing here is that, of course, you have to denature it and have single-stranded molecules so that when you hybridize them, they become double-stranded. So all of that is embedded in the protocol. So if your gene is binding to this array from this particular individual, it will fluoresce red, right? Well, just this solid red. If it's green, it will fluoresce green, right? If it's present in both, then you have the yellow, right? And that's the overlapping between the two colors. So you can tell, is this gene only present in this sample? Is this gene, I'm looking at green, I hope this is green, present only in this sample, or is it present in both, right? So you can tell is your gene expressed or not. Yeah, and this is really, after you apply this, you have this small region. Again, this is representing 676 genes. You run it through a machine. The machine detects the signal and gives you a reading and tell you this is the intensity of this particular signal. And this is the gene that's associated with that spot. So that's your gene that you're interested in. Okay. So you can use this technology, and this was widely used and it's still widely used depending on the group. And once you have all of this information, you need to find a way to go from looking at this chip, because this is a blown up, imagine this is a blown up version of what you see. Just looking at this is completely useless, right? It doesn't give you any information besides the fact that yes, your genes are expressed in one sample or in another sample or in both samples, right? That's really all it's telling you from just looking at this. So you need to have a way to visualize the expression of each gene and the expression level. How much is this gene expressed? So the way that we visualize transcriptomic data, or one of the most common ways we visualize transcriptomic data, is using what we refer to as heat maps. Yeah. So these are maps which generate some kind of, looks like it's generating some kind of a signal. So they're referred to as heat maps. And in these heat maps, there's lots of information about the gene, the condition, and how similar one gene is to a next gene, and also the intensity, how much of that gene is expressed. In order to get from this level to this level requires a lot of computational matrices and statistical analysis that takes into account a lot of information provided in this sample. So it takes into account things like, is there a background signal that's caused, like, skewing your real signal, right? How is there regions within that sample where there is no signal coming from it, but then when you average everything, it's averaging it against this one spot that shouldn't be there. So you do a lot of different types of clustering analysis to be able to get this final heat map. Depending on the question you have, you choose that clustering method, okay? Depending on your lab group, you choose the color you want to use. So different groups use different colors, okay? If many groups and publications and editors or publication journals prefer for you to not use red and green, just because some people have difficulty really seeing the difference in these two colors. So people use a range of different colors, and that's really all what these different colors mean. The information provided is usually very similar. So you have to tell people, okay, if it's really red, that means that gene is expressed very, very low. And that's usually represented as a minus number. If the gene is highly expressed, highly abundant, it will be a different color that's super bright. And you tell the readers or the viewers that this is 3, this level is 3. And again, these are numbers that are determined based on particular types of analyses, yeah. So you can determine it. Some people might say they prefer red to be something that's highly expressed and green to be something that's done. It just depends on the users. But these are kind of typical ways that you can view a heat map. What's also important in all of these heat maps that's shared, the rows are always representing genes. So any heat map you see, if you go to pull up a paper right now, or you go on a website, if you see a heat map, the rows should always represent genes unless they tell you otherwise, okay? So each of these rows, each of these really skinny lines, which you cannot see, represents a single gene, okay? And these are names of the gene that you see on the right. Some people write it, some people don't. On the left, what you're seeing is a similarity, right? So it's kind of a phylogenetic. So the ones that are black means there's no expression, yeah? Or the ones that there's no change in expression, yeah? So it's kind of a net value. And the same with the ones that are white here, right? There's no change, yeah. So all that information, because you're looking at all of the genes in the genome, has to be provided, right? So it's not like you're cherry picking only the ones that are upregulated or highly expressed or the ones that are loaded. You have to put everything that's there. And some genes are just not going to be expressed, and you know why that is, right? Because not all genes are expressed at the same time in certain conditions or certain cell types, right? So you're going to have genes that are not expressed at all, yeah. And you can do similarity based on phylogeny, right? So you have a phylogenetic tree, which not sure how much of this you guys are familiar with, but you have this phylogenetic tree, meaning that these things are shared based on similarity in sequences or evolution, right? But it just depends. Here, in this case, this phylogenetic tree here, where you're clustering these genes next to each other, are more likely based on something you provided. Like you cluster it based on gene families or certain types of processes that these genes might be involved in. And that was just what's determined here. And then in your columns are usually your samples, what they are, maybe the treatment, maybe the developmental stage, right? So if you were looking here, you would say, okay, these 3 columns that are present here represent a particular genotype and condition, because most of these experiments, everything we do, we have to do 3 times for anyone to believe us. So there's always 3 biological replicates, and you should do at least 3 biological replicates, meaning that you repeated this experiment at least 3 times. So people might choose to show for every time that I've repeated that experiment, this is how similar it is, right? And this is really beautiful because everything is really similar here. Maybe some people prefer not to, so they average those 3 biological replicates and just showed really just 1, right? And that's why this is different. Sample 1, sample 2, sample 3, because you're not comparing the one next to each other. You're comparing everything that's within one. Well, so this is really how you depict these maps. So the problem with microarray, or one of the so I'll tell you what some of the advantages of using a microarray is. It's cheaper than using RNA sequencing. It's easy to use, right? Because you can just call the company and say, I would like to buy this chip. Could you send it for this particular genome? Could you just send it to me? So you can do it. The problem with, or one of the challenges with microarrays is that it doesn't represent all of the possible expressed regions of a genome, okay? So if you have a gene that is alternatively spliced, it will not be represented on that microarray. All of the possible splice isoforms is not going to be represented, right? It's just going to be the one that is expressed when you collected that DNA sample and put it there, right? And actually, it's not even going to be the one it's going to be what's encoded in that DNA, right? And you're going to assume that that gene is going to be properly spliced for this transcript, but you know that alternative splicing happened. So maybe your gene, when you isolate your mRNA, is a different version, and that version is not going to be represented on your microarray. The other thing that might not be represented on the microarray are small RNAs, right? Or microRNAs might not be present because these are messenger RNAs that there's no real DNA that's really encoding it that you can say, okay, this is the DNA for that, yeah. Because these are usually generated post-transcriptionally and regulated post-transcriptionally as well. So you'll miss out on that if you have a novel isoform, for example, something new that you want to discover, microarrays usually don't represent that, right? It really just represent what you isolated, what is very basic within that genome, but it is cheaper, yeah? So RNA sequencing is way more powerful in that sense because it gives you a more precise measurement of not only how much transcript is present in the genome, but what other versions of that transcript is present within the genome. And for that reason, it's become the popular method of choice. It's definitely way more expensive to use. If you want to because to sequence one run of an RNA-seq experiment, it's about $3,000. Yeah, so it's a little bit more expensive, and this is just on average. There's a lot of stuff that goes along with that, but let's imagine that you want to do a sequencing reaction. You want to budget about $3,000 just for the sequencing reaction. And then there's a whole lot of other costs that's associated with getting you to the point where you can actually sequence it. So this method, again, total RNA, you're isolating messenger RNA with your oligo-DT primers, right? Because you're isolating poly A tail. You can also do this with other types of small RNAs and just don't use this oligo-DT, right? You can isolate all of the possible RNA versions in the genome by using other types of primers or ways to isolate those RNAs. But in this case, you're a messenger RNA, you're isolating the messenger RNA. Because they're a messenger RNA, they're going to be long. You need to fragment them, you want to make them shorter because they need to be shorter for you to sequence them. Then you're going to do your cDNA synthesis, like what we discussed with making cDNAs, right? You're going to do your cDNA synthesis, your second strand cDNA synthesis. You want to repair the ends, right? Because you don't want the ends to be degraded and chipped away with whatever nucleus you might have in the solution. So you repair them. You add A to the ends of the 3' end to keep them stable. And then you put adapters to both ends to protect them as well. And the adapters are also, if you remember from the cDNA synthesis, you're adding adapters that will be used as the primer region to add a primer. The adapter you're adding, you know the adapter you're adding, right? This red piece is what you designed, you've ordered it, you know the sequence. So when you put the adapter, then you can design a primer to these adapters because you know the adapter, so you can design the primer for it. And these adapters will be added to all these pieces of DNA, and then you can just use PCR amplification. Make sure you have enough, right? So you amplify it. This PCR amplification step is very important. And actually critical for sequencing because you want to make sure you have enough RNA to run in the sequencing machine. You sequence it, and it will give you back these short reads. What's different with this compared to the Sanger sequencing method for genome sequencing? I told you guys that you do 600 to 800 base pair sequencing, or even the shotgun sequencing is usually, I don't know, couple hundred, could be 100 base pairs as well. With these RNA sequencing, you can have about 75, again, depending on cost, 75 base pair sequences. So it's a lot of little sequences that you need to now align back to a genome. So you need to have a reference genome, meaning that you need to have a genome that's already been sequenced to be able to do this. Okay, so then you'll take these little reads, and because they're matching pieces in the genome, you can put them together and you can say, okay, this gene is highly expressed because you have a lot of sequences to it. And a lot of these sequences came from the RNA that you isolated. Compared to here, where you have maybe just one sequence that's added here. So this region is probably not highly expressed because there's only one read that's representing it, right? So you can look and see in the region in the genome which genes are highly expressed, and you can quantify this so you know how high it is using different methods. Okay, so it's a beautiful technique. I mean, and it's used quite often to really compare how the entire genome is changing in terms of the expression across different developmental stages in response to different chemicals, different times of day. So it's really a powerful method to doing that.
But how come is it quantitative? I don't see any step where.
So, right, so this doesn't show you the quantitative part, so you can quantify this after, yeah? So after you get to this step and this is just mapping it to the genome. After this step, you have to use bioinformatics and lots of computational methods to determine, A, is your gene really differentially expressed compared to just any other gene that's at a normal level in the genome? Is your gene, is the expression or the mapping of these reads, do they represent something that's highly expressed compared to something else? And what is a highly expressed gene? How many reads are you supposed to have to say something is highly expressed? So you really have to go through a lot of other types of analysis to determine what these reads that are mapped represent, right? So usually you quantify this, you have a scale, you're comparing across different scales. So there's many different ways, many different things you have to do after that to really quantify what's happening. Yeah, and also using a statistical method to say whether or not it's actually significant or not, those changes. Because of the way that this method is developed, you can use quantitative PCR on a single gene later. You can't use quantitative PCR on the entire genome, right? Because you will need to know every transcript in that genome that represents every gene and design the primers for everything. So say you finish, you go through all of this analysis, the methods, the statistical analysis and bioinformatic approaches that are used to analyze this RNA sequencing information is really robust and really powerful and really good at predicting very good at quantifying the level of expression. So you can do that, and that's usually what people do. After that, you can still go, if you say, okay, after all of this analysis, I have 5 genes that seem super interesting. I want to make sure that they're really what they are. Then you can do qPCR and really quantify only those 5 genes. Yeah, so that is done quite often. So different groups do that. There's right now discussion in the field on whether doing a qPCR after RNA-seq is really important, but it really depends on the question and who and what you're trying to address and what you're trying to claim as well, right? If you're trying to make a big claim, then you want to have other ways to validate your claim, right? So then, doing a qPCR will be a no-brainer to do after, but this is just showing you once you map the reads. But beyond this point, which I don't have detail here, you have a lot of other types of analysis that needs to be done to really understand what this RNA-seq means. So that's a good question, thanks. So that's for expression. So we also do large-scale methods to look at whether or not a gene binds to DNA, so a transcription factor binding to a DNA sequence, or certain types of DNA binding proteins binding to DNA. We use methods to figure out whether or not the histone protein is, whether or not DNA is wrapped around the histone protein or associated with a particular DNA. We discussed the small scale methods, which you guys are really good at now, right? Your filter binding assay, gel shape EMSA, yeast hybrid assay. What is actually missing here is the DNA's footprinting, but all of these methods to determine whether or not your protein is bound to your DNA. There's a large scale method that we use if we want to see, is this protein binding to all, or what DNA in the genome this one protein is binding to? Is it binding to 500 genes? Is it binding to 1,000 genes? Right, so you need to do a large scale method for this. And this large-scale method is referred to as chromatin immunoprecipitation, or CHIP. And depending on what you do after that initial CHIP, determine the outcome of what you want. So after your initial CHIP, you can then combine that with a microarray experiment, okay? Because this will tell you where it's binding and how are those genes expressed. You can do chip sequencing where you isolate where it's binding and then you're just sequencing those regions where it's binding so you know what it is. Yeah. Depending on what your question is, we'll decide which of these techniques to use. The reason it's called chromatin is because you're actually isolating chromatin, right? You're taking the organism, isolating the nuclei because in your nucleus, because that's where all of your DNA is, right? And you're isolating the chromosomes, the chromatin, so the proteins that are bound to that DNA, etc. So that's what you're isolating. So that's why it's called chromatin immunoprecipitation. And it's immunoprecipitation because you need to use a tag, right? You need to use a epitope tag that can be recognized by a specific antibody. This is very similar to the method we discussed when we were discussing how they knew that RNA polymerase had all of these subunits, right? 1 to 12 or 13 or whatever, right? Where they isolated the protein and ran this gel and you saw the different bands, if you guys can recall, right? So it's the same method, right? So you have your chromatin, you isolate it with the proteins bound to it, you chop it up into little pieces, you use a specific antibody to bind, that recognizes these proteins to separate these different fragments. Once you separate these different fragments bound to this protein, you can just get the DNA only and you can sequence it to see what it is. Or you can hybridize it so you can sequence it to see what it is and where it maps in the genome. Or you can put it onto a microarray and see which regions in that microarray it's binding to. Okay, so you can do this method, which is it's a very beautiful method to look again. It's all genome wide, so of course it's costly and intensive and everything. So it just depends on what question you have that you want to use these methods. Ribosome profiling, so you can get so you got an arrow to chip sequencing because this is the step where you would sequence it rather and not, if you weren't going to do chip-seq, you would use this step after you isolated the DNA and sequence, or you go down here and do the hybridization to the microarray. So this is like, so we know we can do genome sequencing for looking at all the genes. We can do transcriptomics, right, to look at all the transcripts that are present and what genes are expressed. We talked about the fact that you make your messenger RNA, it goes through all of this proofreading and taking care of and making sure that it's perfect. And only a subset of those mRNAs are going to be translated, right? The rest are going to be degraded because they're not perfect, right? We can determine which of those mRNAs are being translated using this technique called ribosome profiling, because we can isolate the ribosomes. The ribosomes are going to be attached to the mRNAs. So that means that it has to be actively translating right now. Yeah. So it's very similar in the technique to kind of a DNase footprinting or RNase protection assay, right? So you're digesting the mRNA, isolating the ribosomes. So you get rid of the ribosomes by adding a particular enzyme. And then you can remove the RNA, convert the RNA to DNA, cDNA, and then you sequence it. So you know which mRNAs were being isolated, that were being translated. And you can also map them along the genome so you can see the regions in the genome that's being actively translated. Yeah. Another very powerful method to looking at translation and what's being translated, right? Because again, along all of these steps, many things can go wrong, right? So if you want to feel like, oh, my gene is transcribed, oh, my gene is translated, right? And then now my gene has been translated, my protein now is functional and it's expressed. And that's where looking at protein abundance becomes important, right? So you need to be able to detect the protein. And the way that you detect the protein is using a Western blot, very similar to Northern and Southern blots. However, you're looking at protein abundance. So you have your protein, you run this gel with your protein extracts, and then you would hybridize it to a membrane, and then you use an antibody to your protein of interest to detect whether or not that protein is present or not, and whether it's expressed or not. If you see a band, it's expressed. There's no question about that, okay? So this is what the Western blot is very highly used for. Is your protein functional? Can you detect that protein and how much of the protein is there? Because you can also quantify how much protein is there, because you also have controls that you also run on this gel at the same time. And also a ladder, so you make sure it's the correct size protein that you're looking at, okay? And this is if you were to compare those methods, very similar. Sorry that we're going over just a few minutes. I hope that's okay. If you have to go, I completely understand, but I'll keep going. So it's very similar to what you saw with Northern and Southern blot, right? So knowing the difference between these three is important, right? And what each of them, the information each of these techniques will give you, right? But the idea is very, very similar. Whether you're looking at DNA, is your DNA, your fragment present, how much is present within the genome, RNA, Northern blots, how much is expressed within the genome, and then your protein, is it present, is it functional, how much do you have? Yeah. So very similar idea. And you can imagine that these three can be three small scale methods, right? Because you're doing a Southern blot for looking at just DNA, or just a piece of DNA or a single gene. You're doing a Northern blot to look at a particular gene as well. And you're doing Western blot, you're looking at a particular protein. Large scale method, you sequence the whole genome. Large scale method here, you will do microarray or RNA sequencing. Large scale method here, which we did not talk about to look at protein abundance, is proteomics. Yeah, so you can isolate all of the protein in the genome and determine what they are, okay? So those are the three methods where I want to end. And just to say again, the summary of this lecture, you know, understanding what is an ORF, two approaches to genome sequencing, you know, what are the large scale methods to monitor gene expression. You don't need to know all of the details about the two approaches for genome sequencing, but the idea, one is sequencing first, then mapping later, or mapping first and sequencing later. That's really the extent to what you need to know for those approaches. Large scale genome sequencing method to monitor gene expression, knowing the difference between a microarray versus RNA sequencing. The large scale method, genome-wide methods to detect target sites, right? ChIP-seq versus ChIP-chip. And also the method to detect protein abundance, which is Western blot. Knowing the difference between, again, large scale versus small scale methods is good, because you could imagine you would need to compare and contrast something like that. That is where I want to stop. I did have some poll questions. I can just maybe post them as just practice questions for you guys to go through, since I obviously didn't get to them. Or I can do them here. If you want to stick around and answer, you guys tell me. Otherwise, I'll let you guys go. Tomorrow we will have, I'll post them as some poll questions, okay. And some, okay, yes, polls here, okay, I'll do some of them here, okay, and I'll post the rest for people who have to leave. Tomorrow, again, during office hours, I'll be there at 3. I'll go over again specifically what you guys should be thinking about for exam, for lectures 1 to 11. We'll go over things that I think you should really also focus on the last set of lectures. I know there's a lot of material for the different lectures that you need to go through. Compared to, I think, the first set of lectures, I feel like this is really, unfortunately, the last set of lectures, a lot of memorization, which I don't really like, but it's just the nature of the material. But I'll try to maybe go over the type of experimental questions that you could possibly have available. Yeah. Okay, so I will launch this poll and people can look at it and we can go through this. I'll stick around, until 2. You can go through them and then I'll try to post them along with the study guide for lecture 16, or a little bit more detail of the study guide for lecture 16 by the end of today. And I can answer questions as well if you have any questions. Sorry, I went over, yeah.
Wait, sorry.
Thank you, Professor.
You're welcome.
Oh, Professor, for these questions that you're going over right now, will those ones also be posted?
So yeah, so these poll questions, because some people asked to post it, so I'll post it. So I said, if it's just up to you, someone said just post it right now, so you can stick around and do them, but I'll post them later. Yeah.
Got it. Thank you.
Thank you, Professor.
No problem. Thank you, guys. Bye. Maybe I just do this one since it's only 12 of you. So this is correct, right? I mean, let me launch and share the results for people who are here. So yeah, so I mean, we talked about, if you're looking, because your genome is always going to be the same, right? Whether it's in the liver cell, brain cell, from the same human at the same moment, right? This is the only thing that's going to be the same as your genome. Your transcriptome might change, right? Depending on whether those genes are specific to the liver cell that needs to be expressed, or whether or not it's specific to the brain cell, right? The proteome also is going to change because the transcriptome is going to change. Interactome, we didn't talk about interactome, but this is how proteins interact with each other, and they do. So protein-protein interactions are called interactome. We didn't talk about that, so obviously that's not the answer. Okay, so but the genome is correct. But I'll post these online with the correct answers as well a little bit later today. And the other questions as well, because I have two other questions. So I'll let you guys go. I'm still here for another 2 or 3 minutes if you have any questions.
Professor, would it be possible to record tomorrow's session for office hour since you're doing an exam review?
I can try to record it, and I hope that I remember. I can try to record it. Yeah, I can try to record it. I mean,
Please.
For particular questions people are asking, depending on what the format. So when I'm going over the review part, I'll record that. But if people are asking questions, I'm not going to record that. Like the part where people ask questions. Yeah,
That's totally fine. Yeah, because I have a presentation at the same time tomorrow. So I won't be able to make it. Thank you so much.
Okay. Yes, I mean, to answer that question, Sumama, I just don't feel comfortable recording students. I mean, you guys might be fine with it, but I don't feel comfortable. So I'll record my part when I'm talking, but if there's a lot of questions from students about particular things in the exam that they might be struggling with or whatever, I prefer not to record that. I mean, it might be difficult to separate the two, but I'll try, or I can go back after the fact and separate it. So yeah.